Hello, good afternoon. I hope this is on and I hope you can all hear us. Before we begin, we would like to acknowledge that we gather here today on stolen lands, the land of the Wurundjeri people, of the Kulin Nation, and pay our respect to elders past, present and future. We'd like to acknowledge that sovereignty was never ceded, and this always was, always is and always will be Aboriginal land. Okay, thank you so much, and can we please welcome the wonderful Professor Sharon Lewin to this HIV Cure Chat. Hello, Sharon.
Hi, Nick. Very good to be here.
Thank you. For those of you who might not know, for some crazy reason, Professor Sharon Lewin is the founding director of the Doherty Institute, and we're going live from her office, which has so graciously opened the doors for us today. Sharon was also the co-chair for AIDS 2014 here in Melbourne. And she is, without a doubt, one of the world's leading minds on HIV cure. So we are very lucky to have her in Australia and chatting with us today. Sharon, thank you so much for coming in to have a chat, or letting us come in to have a chat, rather.
Welcome anytime.
You've recently just come back from CROI in Seattle, haven't you?
Yeah.
Tell us a little bit around what CROI is and what was happening there.
So CROI is the big annual American meeting for everyone that works in HIV. It's heavily science focused. It's a bit different to the meeting that we hosted here in Melbourne, which had community and political leaders and sort of much more diverse. CROI is very science oriented, and it has the latest science basically in the clinical world, in the basic science world, in the prevention world. It was in Seattle. I think it was the 20th meeting of CROI, and in fact I've been there every year since for that last 15 years, so it's always a great meeting because the latest science is presented there. 3.5 days of scientific presentations, lots of meetings and some exciting stuff.
Yeah. Lots of very exciting stuff for scientifically minded people like yourself.
Yeah.
For those of us who are very passionate about HIV because we live with it, because we're affected by it, because someone we love is possibly affected by it, we can often get bogged down in the science of HIV. And so we thought with this chat what we're going to do is we will talk through some really fundamental basics of HIV cure and the science around that.
Yeah.
And then we'll get to some specifics on where we're at in 2017 with some really exciting updates that have happened from CROI.
Yeah.
Is that correct?
Yeah.
Great.
Sure.
Well, the first question I want to start with, Sharon, and it comes up quite often in TIM and whenever I talk to someone about HIV, is that there is a myth, shall we say, or a conspiracy theory that the cure is out there, it's already been discovered, and it's just that pharmaceutical companies are sitting on that cure. Sharon, are you sitting on the HIV cure? Are you in the deep pockets of Big Pharma?
I would love to be sitting on the HIV cure. It's not there.
We will check under this chair. We are in her office, so I will check all of these chairs.
It is not here. I wish it was. I hear it a little bit too, actually, even amongst doctors or skeptics that are always fearful of Big Pharma. Pharma's got a mixed reputation, but Pharma did deliver antivirals for HIV. Pharma have done some fantastic things. Engagement of pharma into the search for a cure is really important, and it's a big part of the work that I do together with the International AIDS Society. We unfortunately don't have a cure, they don't have a cure, but there are many big pharmaceutical companies working on this. Not everyone, and probably not at the level of investment that there has been in other areas, and that's not because pharma don't want a cure. I think it's because it's still in a very high risk stage because we don't have something yet that we can develop, but no, unfortunately there is none. I'm not sitting on one. I'm working really hard. I'd love to see one, but I think we're a few years away. And
so the majority then of the hard work, the grunt research that's taking place is happening in institutions like this, more so than pharmaceutical companies. Would that be right?
Yeah, I mean, academia is kind of where new ideas come from, more risky research, what we call discovery research, trying to find a lead that will lead us to a cure, and then pharma takes over once they see something that's promising, and then they develop it into a drug. And there is a bit of discovery research happening amongst some pharmaceutical companies, actually a lot of, especially mainly two companies, Gilead and Merck, but other companies are doing discovery research. But they often, pharma really gets involved when they see that it's a drug that they can take to market.
Okay, and is that then wrestled off research institutes like this one? Is there a point where the knowledge gets passed over, or is it a bit of a race to cure, to find that cure?
Yeah, it's, science is based on intense competition, but also intense collaboration. It's an interesting mix. If you really have to be collaborating with lots of different scientists and clinicians and community in order to get advances in science. Big believer in collaboration. On the other side, science is also intensely competitive. Where industry or pharma come in is if you have something that you think is going to be commercialised. We're a long way off that at the moment. I would say though that there are lots of initiatives to get more collaboration. So I'm part of a very big network of collaborators we call DARE, the Delaney AIDS Research Enterprise to Find a Cure. So DARE to Find a Cure. Delaney, named after Martin Delaney, who was a really well-known activist for HIV in the US, highly effective person, and this program is named after him, and it's funded by the American government at the moment, but it is funded, National Institutes of Health, and they specifically want people to collaborate and also to bring in private industry. So in our big, what we call a co-laboratory, lots of people, collaborators, we've got investigators from Australia, myself and my team here in Melbourne, a group from Sydney headed by Sarah Palmer, a group in San Francisco and across the US. But we also have lots of industry partners, so Merck is part of this, Gilead is part of it, small and other big pharma. So a lot of the funders now are saying you have to work together and you have to work with industry to really move this ahead.
Yeah, right. How fascinating. For those watching, Merck, when we talk about the pharma companies, Merck is the pharmaceutical company who discovered combination therapies. Was that right? Correct to say?
Merck was probably best known. They've been developing a lot of antiviral therapy, but they've developed integrase inhibitors and raltegravir, which was really a great advance in one of the better antivirals around.
Fantastic. Now, you've mentioned time. You've mentioned that it's the cure is down the track, and we don't really know when. You must get asked this all the time, but are we talking 10 years? Are we talking 20 years? Are we talking 50 years? Do we just have absolutely no idea? Tomorrow could come the breakthrough, and then we roll on from there?
Yeah, I really hesitate to answer that question because it always comes back to who. And I think at the moment it's unknown, but in fact, often when there's major discoveries in research, things can just turn around very quickly. I don't want to say that to raise false hope, but if you look at hepatitis C, for example, where we now do have a cure for hepatitis C, we have drugs that are being rolled out across Australia. 10 years ago, we didn't even know how to grow hepatitis C in a test tube model. So sometimes you just need something that can dramatically change the field. So in the current pace of development, I think finding a cure we know is going to be really difficult. There have been some advances in our understanding. There's some people that can naturally control the virus off treatment, something which we call remission, not truly cured. You can still see the virus there, but it's at very low levels without medications. I think we'll see more and more cases of what we call remission, but I think a cure is still going to be a long way off. It's not going to be an easy road. And
if that breakthrough moment that we can't predict when it's coming, when that does come, what would be the kind of standard timeline, do you think? Because it has to be tested, has to be proven, has to then be approved, and then produced and put into the market. So what's that? I mean, if the breakthrough moment happened tomorrow, for example, we still have to go through a lot of regulatory steps. And so, at a minimum or maximum, what kind of time frame are we looking at there?
Very, very hard to say. Depends what it is. Again, if I go back to hepatitis C, from the initial discoveries of these antivirals to proving that they cured hep C, this was 5 years. There wasn't a huge. That to me is seems very, very quick. And then now licensing the drugs, funding the drugs in a country like Australia. So it can move. Things can move very quickly. But a cure could be for HIV is a lot more complex than hepatitis C. So hepatitis C is also another virus that I'm sure many of your people that listen, that subscribe to Tim, know about. It's a sort of similar family of viruses, but it's got one key difference, and that is that it doesn't become part of the person's DNA, and that's why it's has been much easier to cure hepatitis C as opposed to HIV. So HIV's got some very unique problems, but you know, we've had some major advances already, many major advances in the history of HIV. So I'm optimistic that we're going to be able to advance this one as well.
Absolutely. Well, we need to be optimistic, don't we? Let's talk about some of those problems, though, because it is a very specific science, and that's not going to come as a shock to anyone who's actually a scientist. But when it comes to HIV cure, what we're dealing with, the main hurdle to be overcome, as I understand it, are the latent reservoirs. Is that correct?
That's right,
yes.
yeah.
Explain a little bit to our viewers what the latent reservoir is.
So when HIV infects someone, it infects certain cells and it replicates like mad in people that are not on treatment. And when you put people on treatment, all of that replication gets dampened down and stopped. But the virus also can go into hiding at the same time. That's what we mean by latency or a latent reservoir. The virus goes into hiding where it burrows inside the person's DNA. Very few viruses go into hiding. We estimate about 1,000 cells in someone with HIV on treatment. And the virus that goes into hiding, which is the latent reservoir, doesn't cause any trouble for someone that's on treatment, because the treatment is controlling all the virus that normally is replicating, and it's cleared out on treatment. But as soon as someone stops treatment, that virus that's in hiding can pop out at any time, just randomly actually. It pops out. They estimate the virus pops out sort of once every 6 days. But in someone that's not on treatment, the virus can then take hold and then get going again. So as soon as someone stops treatment, usually about 2 to 3 weeks, and by that stage one of the virus is in hiding, has popped out, and then the whole thing gets going again. And so we know that the virus can go into hiding, forms this latent reservoir. So we need to get rid of the virus that goes into hiding. And we also, there's been a bit of a shift now to thinking that you really also need to beef up the immune system at the same time, because it's unlikely that you'll get every single virus that goes into hiding. But if you beef up the immune system's armed and ready if any virus pops up. So there's two arms of cure research: getting rid of the latent virus and at the same time building up someone's immunity against the virus.
Oh, okay. So that's actually separate sort of thing. Oh, we have a question. Yay. Question is, why can't people choose to undergo what was done to the Berlin patient? Oh, this is a really great question. Thank you, questioner. That's fantastic. I think, Sharon, do you want to? Bit of background, sorry, firstly. The Berlin patient is currently the only person who was living with HIV to be cured.
Yes.
10 years ago.
Yes, it was his 10-year anniversary on February the 7th, actually. They had a big party for him.
Did they?
Yeah, in fact at Croy there was a community day around cure, and the Berlin patient's name is Timothy Brown. He actually prefers to be called Timothy Brown. He didn't reveal his identity at the beginning and was the Berlin patient, but now he actually says he prefers to be called Timothy Brown. And there was a big birthday party for him for his 10-year anniversary, as I said, February the 7th. So he is, as you say, the only person cured of HIV, meaning that there is no HIV detectable in his body at the moment, and he's been off treatment now for 10 years.
Yeah.
But he had a very unusual situation. He had HIV. He was living in Berlin at the time. He is American, but was living in Berlin at the time and developed a very nasty form of blood cancer called leukemia. And for treatment of his leukemia, he needed a bone marrow transplant, which is sometimes what we do in bad cancers, where you destroy the person's bone marrow and you give them a new bone marrow that doesn't have the cancer. And his doctor at the time thought, well, what if I gave Timothy a new bone marrow that was resistant to HIV? And we know that about 1% of people are resistant to HIV because they don't have the receptor that HIV needs to get inside cells, and that receptor is called CCR5. So this clever doctor, Gero Hooter, happened to look hard for a bone marrow donor who matched everything for Timothy, but also carried this mutation in CCR5. So Timothy Brown received this bone marrow from a donor that was naturally resistant to HIV, his drugs were stopped about a day after the transplantation, and the virus never came back. So there's a few things there. In order, when you give someone a new bone marrow, you actually do lots of things to them, because you've got to get rid of the old bone marrow. You give them all sorts of drugs to suppress their immune system. So we don't know whether what cured Timothy Brown was this special bone marrow for someone that was resistant to HIV, or all the other stuff that was done to him for the transplant. Now, the reason why we can't do that for people, even if they wanted to have that done, is that that whole procedure of a new bone marrow is actually a procedure that's associated with an enormous amount of risk. There's about 25% risk of death with that procedure. He, of course, had it because his leukemia had a 100% chance of death. So we take those risks in people that are very, very sick. But in someone living with HIV who's taking antiviral therapy, very different situation. Yeah. But what Timothy Brown has told us, what Timothy Brown has taught us, is that maybe we can do something a bit more safely, similar to what happened to him, but not requiring a bone marrow transplant.
Because it's quite invasive, isn't it?
Very invasive.
I mean, as an HIV positive person, I wouldn't just pop on down
and get
to
a
my
bone
local
marrow
hospital to get a
transplant.
bone marrow transplant.
I don't think anyone would do it. Would anyone would do it because they would say, look, you're doing really well in treatment. You have a normal life expectancy. And this has got a 1 in 4 chance of dying from this procedure.
Now, on that, and that's another question, is it true that there was another Berlin patient? And that is true that this process has been attempted since?
Yeah, it has been attempted since. In fact, there's 7 reported cases of a similar sort of transplantation, and 1 or 2 others that haven't yet reached the literature but have been presented at conferences. And in most of the other cases, people have not done as well as Timothy Brown because, as I said, there's a 25% chance of risk. So often these people have died in that first year after transplant of infection or the cancer coming back. And no one has been able to replicate this so far. And there might be a whole lot of reasons why, but we haven't been able to replicate it so far. But what is interesting, and I think we'll learn a lot, is that there is a really great group in Seattle that are focused primarily on gene therapy and bone marrow transplantation. They've managed to establish something like this in a monkey model, which means that we can manipulate the system a lot more. But in people with leukemia, all in other types of blood cancers who have then had transplantations from a donor that's naturally resistant to HIV, they haven't done well so far, and we're still waiting for another case of like Timothy Brown.
It's an extraordinary example of what has been core to HIV science research, which is HIV positive people putting their bodies on the line and volunteering for trials, for tests, for research.
Yeah.
And this goes all the way back to the very initial days where people living with HIV, people living with AIDS in those days had a very, shall we say, grim future or lack of a future ahead of them, and they still, even though they knew that the treatments they were going to try and work weren't going to save their lives, they knew that they would be part of helping future generations, and I'm one of those people, so I'm very grateful for those elders who literally gave their lives in the name of HIV science. That obviously was a time now long past, and we get to take our antiretrovirals every day if we're privileged enough to live in a country where we can access them. What can HIV positive people expect if they do put their hands up and they put their bodies on the line for cure research and cure trials today?
Yeah. Well, actually, it's similar, but it's different. So first of all, I said that we wouldn't have antivirals today without Big Pharma. Well, we wouldn't have antivirals today without the courage and activism and participation of people living with HIV. There's just no doubt about that. Not only just in participating in research, but being activists and pushing pharma to really work on this. So there's no doubt that people living with HIV played a major role in where we got to today, as you said. At the time, participating in clinical trials was a bit of life and death for people, but yet still people were did amazing things and participated in research. is a little different with cure research because people are doing extremely well and living really well and active and healthy lives. And so participating in cure research is really around altruism to advance the field. Now many people do this in many other areas as well. So what can we pick? And there are all different types of clinical trials on currently in Australia and internationally. Some clinical trials just require donating blood or maybe tissue so that people can better understand latency, ways that we can get rid of latency or stop it being established, and that is a really valuable contribution. So we have a clinical trial running here in Melbourne where people can donate very large amounts of blood through something called leukapheresis, where you can take literally 10 billion cells from someone, and then it's a special machine that replaces all the blood at the same time, so people can literally in 4 hours donate huge amounts of blood and then walk away feeling completely fine. It's pretty incredible, but amazing contribution because of this huge number of cells that we can now find those very rare latently infected cells and manipulate them in the laboratory. So there are those what we call sort of observational studies, which are really, really important. But there are also a number of interventional studies, meaning giving someone living with HIV on treatment an intervention like a new vaccine or a new drug that's going to try and wake up the latent virus, or give them extra antivirals. There's a whole range of different trials that are being done, and what we often explain to participants in those studies are it's very unlikely that you will get a benefit from your participation. There may even be some risks associated, and they're discussed with the person, but this will make a big contribution to our understanding. And that's the sort of conversation we have with people. We're not going to, we're at the stage now where there's really very little that we can say why we think this might work, but we're very upfront with saying this is very unlikely to benefit you personally. These are the possible risks, but we would not be doing the study if we thought this was very dangerous or there were really serious risks. And that's the sort of conversation we have with people.
And in some cases as well, it involves HIV positive people stopping their antiretroviral treatment, their daily treatment, to participate in some of these intervention.
That's right.
Trials.
That's a
big consideration.
exactly. That's in recent. The earlier studies we weren't doing that. The more, because in the end, what we want as an outcome from these studies is for people to be able to stop their treatment and the virus staying under control. Again, what we call HIV remission. So to test whether something's worked, many of these newer studies require a treatment interruption. And treatment interruptions for a long time were thought to be quite dangerous. And then they are quite dangerous if they're not carefully monitored, and if people stop their antivirals when they've had very advanced disease before starting antivirals. So I think treatment interruption studies are definitely safe. But they need a lot of careful and close monitoring. But again, it's a very significant request from someone because you need that very close monitoring, which means often blood tests done twice a week. And as soon as the virus pops back in most of these studies, treatment is restarted. But they kind of become a very important part of cure research to understand whether remission has happened or not.
And I think for those of us who are, I call us third wavers, people diagnosed 2012 or 2008 onwards, I call us the children of the Swiss statement. That can actually be a really big thing to wrap your head around, because we've come of age into our HIV journeys, accepting the science of undetectability, taking our treatments every day, and to think that you might have to, sacrifice that for the greater good and contribute to something. When it feels like the world is only just wrapping its head around the fact that undetectable viral loads work and they're a thing, and we are absolutely not a risk to anyone else, is a big thing. But I do challenge the positive community out there to think about ways that we can step up and contribute in many ways.
There was a very interesting presentation at the conference I was just at last week at CROI, which interviewed people that participated in a treatment interruption study. And the study was done in Thailand, where in fact there have been real leaders in some very exciting cure research, largely around very early diagnosis and early treatment, and then doing treatment interruption studies as part of their interventions. And they did a very interesting study where they interviewed participants into why they got involved and how did they feel when they had to go back on treatment. And there were some amazing statements. One person, did it because they really hoped that this could have been a cure, but they knew that it was likely there wasn't, but there was this hope there was. And they felt that they had still made a very major contribution by having participated and knew that it was done in a safe environment. So it was obviously a group of people that chose to participate in the study already, but it was interesting what motivated them and how they felt in participation.
I want to come back to CROI in a second, but one, I guess, provocation, not really shown for you to answer, but for maybe something for people to think about, is now that PrEP is more widely available in a certain number of countries, increasingly this one, what that might mean for HIV positive people with negative partners whom for taking a treatment break would just not be an option because one of the main reasons we take treatment apart from our own health is to protect our partners. And maybe just a provocation, something to think about, something to comment, or is the idea that now PrEP is around, would more positive people choose to take a treatment break if they're interested in HIV cure research?
Yeah, and that's something we've discussed in designing these studies. We haven't done a treatment interruption study yet in Melbourne, but we have some studies coming up. An interesting study that we'll be doing together with San Francisco and Denmark, which will involve treatment interruption. And we've discussed this about whether PrEP should be made available to someone's partner, which I think should be the case.
So I think it's great. Let's go back to CROI and let's talk about what seems like the most exciting news that came out of there, which is a vaccine development coming from some Spanish researchers. Do you want to tell us a little bit about that?
Yeah. That was I agree, was probably the most interesting study presented there. Also included a treatment interruption, in fact, and had they not had a treatment interruption, we wouldn't have even known it was so exciting. So I'll explain what happened. So it was a small study, and many of these cure studies are small studies. So they just give what we call proof of concept. They sort of give us an idea that this might work, and then it has to be repeated in a bigger study. So it was a small study of 15 people that were living with HIV who had been treated early after infection. So within, I think, an average of 3 months of being infected, they received antiretroviral treatment. And these 15 people were part of a vaccine study, and they received early treatment, and then they got a vaccine. Now, we always think about vaccines as you get them to protect you against getting infected, but there is a whole area of interest in many infectious diseases, also HIV, that you could give a vaccine after someone was infected in order to re-educate their immune system to fight the virus better than the person's own immune system. We call that a therapeutic vaccine, so it's a vaccine for treatment. There aren't many infectious diseases we do treat like that, but people have been looking at this for a long time in HIV. We've never had a successful therapeutic vaccine, I should say.
Right.
But in this Spanish study, the 15 people that started treatment very early received a vaccine while infected, and the vaccine seemed to stimulate pretty good responses to parts of the virus that don't change a lot. And in fact, that study was presented last year, I think, from CROI. And then 3 years later, they called back those same participants and gave them another booster of the vaccine, then gave them a drug to what we call tickle and tease, as you say, or what we used to call shock and kill, or a drug that should flush out the virus from its hiding place, a drug called romidepsin that's been tested before, and then gave another booster of the vaccine. And then they asked the participants to stop antiretroviral treatment. Now, only 13 of the 15 did stop it, so they had a treatment interruption. And 8 of the 13, virus came back straight away, as you'd expect, in 2 to 3 weeks. And in 5, the virus stayed at low levels under control, but bouncing around, not undetectable, but in the low sort of hundreds to thousands at a safe level.
So for anyone watching, like now, undetectable viral load is less than 20.
Less than 20,
But it was measured at 200 for the partner study. So for it to be kind of from 100 to 1,000, where it's not really in what we would call the super infectious kind of world, that's a pretty great.
Yeah, I mean, I think with someone that had a viral load of 100,000, we'd be telling them you're infectious, but much lower, less than if you had 100. It's related to how much virus there is. If you have a million copies, very infectious. I think 100,000, we'd be still telling people you're infectious and your partner. I would be telling them your partner should be on PrEP. But that's a very low level virus that we think is has people will not lose T cells, for example, at that low level of virus. And so the participants have had different levels of follow-up, ranging between 1 and 6 months. So there's some people, of those 5, some of them have been going for 5 or 6 months with this very low level virus.
Wow.
And there's a few questions from this. Would this have happened anyway without the vaccine and romidepsin? Because there was no control group here, people that were just treated early and then their antiviral stopped. But we know from many, many other studies that it's very rare. 1% of people can control their virus after antiretrovirals stopped. Unlikely. Now which part of the treatment was important? The very early vaccine that they had shortly after treatment, or the vaccine they had 3 years later, or the romidepsin, or the second vaccine. So we don't know which part was most important, and we've got to do another study to really work that out. But what the investigators did find was that when they measured the immune response of the person, it had changed after the vaccine, and it had the immune response was directed to parts of the virus that we know don't change too much. So maybe that was really important.
Right.
And I think there's a lot of interest now in the cure world, not just for tickle and teas to push the virus out, but at the same time to find ways to boost the immune system, either through a vaccine or through other drugs that can stimulate the immune system.
But just to clarify, when we talk about a vaccine, that's not practically, it's not applicable to a HIV negative person. You wouldn't give them a vaccine and kind of, because firstly it's hard to test that, isn't it? We can't ethically research giving someone a vaccine, HIV negative person, and then send them off into the world and kind of tell them to see if they get it.
There still is a lot of, there still is a huge interest, even with PrEP and treatment as prevention, putting people on treatment to partner study and blocking transmission through people. There's still a huge, we don't have a vaccine, and the best way to control any infectious disease, including HIV, would be to get a shot in the arm as a baby, and then you're immune from HIV. And there's still a hope that we will get a vaccine. And there are lots of exciting vaccine developments as well. And there are studies of vaccines. In fact, a very big one has just started now in South Africa. And those studies are now designed with PrEP built into them, so that you have to build in your control arm people that are offered PrEP as well. So these studies need to be very large in order to show benefit, but there still is a huge amount of work being, investment and work being done to find a vaccine.
Great, okay. It's very exciting, and it is the kind of incredible tradition that I think HIV has almost above any other infection disease epidemic, etc., that the people in the labs working on the science, struggling to find cures, vaccines, effective treatment, have partnered with the affected community, which is people living with HIV. And for those of you who are just dabbling in your HIV history, it's really worth going back in time and seeing what an effective partnership it's been from the very beginning.
Absolutely.
It's the first time in history that the affected population have centred themselves within the epidemic, and the nothing about us without us cry. And it's an extraordinary example of the scientific community responding to that and listening and welcoming that. And that's where so many of these advances, I think, have come from, is in that partnership between community and research.
Hugely valuable, really important. It means that researchers focus on things that matter to people living with HIV, and it means the studies are designed much better, and as you say, involvement every step of the way, absolutely critical.
Absolutely. Well, I guess we should probably leave it there. Maybe we should give a quick spruik to hivcure.com.au, which is a relatively new website, but I definitely urge everyone to jump on it. There's so much cure news that gets shared, some of it genuinely fake, some of it raising what we in the community would see as a false hope, because it's complex and maybe we can't quite wrap our head around it, and so we turn off. But hivcure.com.au is a partnership between researchers as well as community organisations to cut through all that noise and to only put up news that's true and matters.
And on the website as well, there's a summary of what clinical trials are available across Australia, how people can participate, and a review of the latest news. And we hope in a language that's easily accessible. But if it's not, we want to hear about it. Let us know.
Yeah, do please comment and let us know through Tim or through other channels just like that if it is a bit dense, because even those of us in the activist and community space, we get stuck in the jargon and all the acronyms. Sharon, I'm drowning in acronyms.
Yeah. We've got a dictionary up there, a glossary, but we still need to be told when this is not working. Yeah.
Yes, and so please do keep tuning in, keep keeping your ear to the ground for Cure news because it is something we need to work on as a community. Professor Sharon Lewin, thank you so much for taking the time to let me into your office and chat about HIV Cure today.
Thank you very much. You're welcome anytime.
Oh, thank you. I'm going to finish this right now.